Welcome to this lecture series on eukaryotic gene expression, basics and benefits, and we have been discussing so far about transcription regulation, how it takes place in eukaryotic cells, various general transcription factors and what kind of promoter elements they recognize, how RNA polymerase along with general transcription factors binds and interacts with a number of upstream transcription factors and regulates transcription. And after looking at the various events that actually takes place inside a nucleus, we started looking at how molecules, by interacting either with cell surface receptors or with intranuclear receptors, are going to influence gene expression. So, we had a series of lectures on discussing how molecules which interact with membrane receptors transduce signals inside the nucleus, ultimately leading to the activation of transcription. And in the last class, we discussed about intracellular molecules which diffuse through the cell membrane, bind to intracellular receptors, and then this ligand receptor complex goes inside the nucleus and activates transcription of various genes. As an example, we started discussing about steroid hormone receptors, and in the last class we discussed about hormones like glucocorticoid hormone, mineralocorticoids, estrogens, progesterone, androgens and so on, how these molecules diffuse through the cell membrane, bind to specific intracellular receptors which are present in the cytoplasm, and once the hormone binds to this cytoplasmic receptor, it undergoes a conformational change, and then the hormone receptor complex goes inside the nucleus, binds to specific response elements known as hormone response elements and activates transcription of target genes. So, what we will discuss today is to discuss another group of receptors known as the type 2 receptors, and first we will start discussing about how these type 2 receptors are different from the type 1 receptors or the steroid hormone receptors, and how these receptors were discovered and what kind of response elements these receptors bind and so on and so forth. So, let us start discussing about the regulation of gene expression by type 2 nuclear receptors. As I said in the last class, we discussed about one of the members of the nuclear receptor superfamily, namely the type 1 receptors. The type 1 receptors are characterized by the presence of a domain structure. There are three major functional domains: a hypervariable amino terminal domain, a DNA binding domain or the DBD, which consists of two zinc fingers, and the ligand binding domain, which actually binds a specific ligand. So, in the case of glucocorticoid receptor, the DNA binding domain specifically recognizes the glucocorticoid response elements in the promoters of various target genes, whereas in the case of estrogen receptor, the DNA binding domain will recognize the estrogen response elements of various target genes. The same way, the ligand binding domain of glucocorticoid receptor will bind to glucocorticoid hormone, whereas the ligand binding domain of an estrogen receptor will bind to an estrogen hormone. So, by differential DNA binding as well as differential ligand binding, this group of receptors are able to manifest their physiological receptors by binding to specific response elements and activating transcription in response to specific hormones. So, the type 1 receptors, one of the major characteristic features of type 1 receptors or the steroid hormone receptors are they undergo nuclear translocation upon ligand activation and bind as homodimers to inverted repeat DNA half sites called the hormone response elements or HRE. So, the major distinction between these type 1 receptors and the receptor that we are going to discuss now is that the steroid hormone receptor are primarily present in the cytoplasm, and when the hormone like glucocorticoid hormone or estrogen binds, then it undergoes a conformational change, and the ligand bound receptor then translocates to the nucleus and binds to specific hormone response elements, which primarily consists of inverted repeats. For example, in the case of glucocorticoid receptor, it is AGA, ACA, TGT, TCT. So, it will recognize, it will read the same on opposite strands. The examples that we have discussed under this category are receptors activated by steroid ligands such as glucocorticoid receptor, mineralocorticoid receptor, estrogen receptor, progesterone receptor and androgen receptor. This just shows what is called as an immunofluorescence picture, where you try to localize where the receptor is present in the absence or presence of a hormone, and as you can see here, you use a antibody which is fluorescently tagged to a secondary antibody, and then you look at the hormone receptor both in the absence or presence of the ligand, and you can see in the case of glucocorticoid receptor, when you do such immunofluorescence studies, you can see the glucocorticoid receptor is primarily present in the cytoplasm when there is no hormone. The moment you add the hormone, the receptor translocates to the nucleus, and you can see you no longer see the receptor in the cytoplasm, but all the receptors moved into the nucleus. So, these are one of the hallmark characteristics of type 1 receptors. That means they bind as homodimers to these palindromic sequences. They are normally present in the cytoplasm and then translocate in nucleus when ligand is present. Some of the members of the steroid receptor family that we discussed about are the glucocorticoid receptor, mineralocorticoid receptor, progesterone receptor, androgen receptor and estrogen receptor. There are different isoforms of these receptors, alpha, beta and so on, either because of differential splicing and so on and so forth. This also tells you that the DNA binding domain of all these receptors contain 2 zinc fingers, and you can see there is very high degree of homology in the DNA binding domain of such receptors, whereas the ligand binding domain, the homology is not that much because they bind different ligands. But they all share certain common characteristic feature, and these members that they constitute together the steroid receptor superfamily, which again comes under the nuclear receptor superfamily. And I also discussed the glucocorticoid receptor, mineralocorticoid receptor, progesterone receptor and androgen receptor. They all recognize a sequence called AGA, ACA, 3 nuclear spacing TGT, TCT. It will read the same way, the other opposite stand AGA, ACA, 3 nuclear spacing TGT, TCT. So, they are called as palindromes or inverted repeats. In the case of estrogen receptor, there are only 2 bases which are different, distinguishes an estrogen response element from glucocorticoid response element. And in the case of estrogen response element, instead of AGA, ACA, you have AGG, TCA. That is the only difference. So, today let us not start discussing about what are these type 2 receptors. The type 2 receptors also have the same domain structure as the type 1 receptors by the fact that they also contain a hypervariable amino terminal domain, they also contain a DNA binding domain consist of 2 zinc fingers, and they also contain ligand binding domain to which certain non-steroid hormones go and bind. Certain vitamins and non-steroid hormones bind to the ligand binding domain. But the important characteristic features which distinguish the type 2 receptors from type 1 receptors are, these receptors are often present in the target cell nucleus regardless of the presence of a ligand. So, if the ligand is there or not, the receptor is already present in the nucleus, and usually they bind as heterodimers with another important receptor called as Retinoid X receptor or RXR, and to sequences which are direct repeats. So, there are 3 important points which distinguish the type 2 receptors from type 1 receptors or the steroid receptors. Number 1, they are already present in the nucleus even if then there is no hormone. Number 2, the steroid receptors bind to DNA as homodimers, but these type 2 receptors usually bind as heterodimers, and the heterodimeric partner for all these receptors is a receptor called as Retinoid X receptor or RXR. We will discuss in detail what exactly is this receptor. And the third important difference is that while the steroid hormone receptors bind to palindromic sequence or inverted repeats, these type 2 receptors bind to direct repeat motifs. Some of the examples or classic examples of type 2 receptors are receptors for thyroid hormone, retinoic acid and vitamin D. So, if you now do an immunofluorescence just as the way I showed in the last slide, in the case of steroid hormones, in the absence of hormone you do not see the receptor in the nucleus, they are all present in cytoplasm. When you add a hormone like the glucocorticoid hormone, the glucocorticoid receptor now transfers to the nucleus, whereas in the case of the type 2 receptors like retinoic acid receptor or thyroid hormone receptor or vitamin D receptor, even in the absence of hormone you can localize the receptor in the nucleus. So, these are the 3 major distinguishing features that discriminates the type 2 receptors from the type 1 receptors. Now, let us begin by discussing the type 2 receptors by taking the thyroid hormone receptor as an example and see what led to the discovery of these type 2 receptors and how the knowledge of steroid hormone receptors actually helped to understand some of the major functions of these type 2 receptors. So, the thyroid hormone receptor, what is thyroid hormone and what is its receptor and how was it discovered and what kind of response elements this thyroid hormone receptor goes and binds. The thyroid hormone or thyroxine is actually synthesized from tyrosine. So, tyrosine is a precursor for a number of hormones like epinephrine or thyroxine and so on so forth. Thyroxine basically is nothing but is also known as T4. It contains 4 iodine molecule atoms, and in the body the thyroxine exists either as T4 or triiodothyronine. If you remove one atom of iodine from T4 by enzymes known as deiodinases, the T4 gets converted to T3, and usually the T4 and T3 are present in the ratio of 20:1 inside the cells. So, both coexist, but the T3 is at least 3 times more potent than the T4. So, today when I am going to talk about what are the physiological effects of thyroid hormone, the thyroid hormones which are synthesized by the thyroid gland and the hormone by the thyroid gland performs a number of important functions, some of which are listed here. They play a very important role in metabolic regulation, they increase metabolic rate, they are involved in growth and development, they also increase they have increased catecholamine effects. There are a number of other physiological effects thyroid hormones, but these are some of the major effects. All these effects of thyroid hormones are probably mediated by the thyroid hormone receptors, and let us now discuss and see what are these thyroid hormone receptors and how they were discovered. So, the ligand for thyroid hormone receptor is thyroxine, either T3 or T4. So, let us now start looking at what are the historical links between the type 1 receptors or the steroid hormone receptors and thyroid hormone receptors, because it is the knowledge that we gain from steroid hormone receptor that led to the understanding of the thyroid hormone receptors or the type 2 receptors. Now, I had mentioned in the previous class how with the advent of molecular biology in the early 80s, the advent of cloning techniques, the advent of genomic libraries and cDNA libraries, and the ability to screen these libraries using antibody specific for proteins or using oligonucleotides which were designed based on amino acid sequences really helped to clone the genes coding for the various steroid hormone receptors. And I also mentioned that one of the first hormone receptors which were cloned is the glucocorticoid receptor, which was cloned in the Ron Evans lab and was published in Nature in 1985. And for today's topic, what is most important is that in the same year and in fact the same issue of Nature, the same group also demonstrated that if you look at the domain structure of the glucocorticoid receptor, which contains a amino terminal domain, a DNA binding domain and a ligand binding domain, it was very similar to a oncogene product known as VRBA. For today's topic, this is going to be very relevant. Now, what is this VRBA and why this paper became very important? That is the demonstration that the structure of glucocorticoid receptor is very similar to an oncogene product coded for by an oncovirus like a erythroblastosis virus. The following year, again two groups from Ron Evans lab and from Wenstrom's lab actually demonstrated the cellular homologue or the proto-oncogene of this VRBA is nothing but thyroid hormone receptor. The CRBA gene encodes a thyroid hormone receptor. The CRBA protein is a high affinity receptor for thyroid hormone. These two are the classic papers which actually linked the thyroid hormone receptor on one hand to the steroid receptors and on another hand to cancer. So, let us now examine a little more detail. What is VRBA? Now, VRBA is an oncogene expressed by avian erythroblastosis virus or AEV, which induces erythroleukemia and sarcomas in chickens. Okay, so it's a cancer causing protein. When you introduce this VRBA into cells, the cells become cancerous. But what became clear is that once it was discovered that the VRBA has very high degree of identity or homology to the thyroid hormone receptor, people started asking the question, what is the difference between VRBA and a thyroid hormone receptor, and soon they discovered that VRBA also binds to the same DNA sequence as the thyroid hormone receptor, but it does not bind to hormone. So, the viral oncoprotein VRBA is nothing but a mutant form of thyroid hormone receptor which cannot bind ligand, but it can bind to the same DNA sequence as a thyroid hormone receptor. This knowledge clearly indicated that VRBA is homologous to thyroid hormone receptor, and therefore, it gave a hint that deregulation of thyroid hormone signaling may lead to cancer. So, you can see the cloning of glucocorticoid receptor and the demonstration that the domain structure of glucocorticoid receptor is very similar to a viral oncoprotein, and then the knowledge that the viral RBA gene, and if you look at the cellular RBA gene, there are some differences, and one of the major difference between the VRBA and the cellular VRBA, CRBA is nothing but thyroid hormone receptor, is that while the thyroid hormone receptor can bind to thyroid hormone, the VRBA cannot bind thyroid hormone. So, on one hand you have steroid hormone receptors, you have a thyroid hormone receptor, and you have cancer. So, they found a common link between three different areas of research. This is a very important observation. So, the steroid hormone receptors share the same kind of a domain structure either with VRBA or the thyroid hormone receptors, and although steroid and thyroid hormones are neither structurally nor biosynthetically related, the fact that their receptors share structural homology suggested that there exists a large superfamily of genes whose products are ligand responsive transcription factors. So, the early 1980s actually revolutionized the entire field of nuclear receptors, because till then people thought the mechanism by steroid hormones activate gene expression is different, the mechanism by which thyroid hormones activate gene expression result is different, and nobody thought thyroid hormone may be involved in cancer. But with these 2 or 3 papers that were published, it became very clear that although steroid hormones and thyroid hormones are not structurally similar, they all seem to be binding to the same kind of receptor molecules, and therefore, the mechanism by which they are activating expression of target genes may be very similar or identical. And this also suggested that not only thyroid hormone receptor and glucocorticoid receptor, there could be many other receptor molecules, and therefore, all these receptor molecules may actually constitute a superfamily of nuclear receptors. So, this was a conceptually, this was a very important advancement in the area of transcription factors. So, it was proposed that there exists a steroid thyroid receptor superfamily of proteins. So, this was a classic review. This was a classic review which was published in Science by Ron Evans, who really proposed that there exists a steroid thyroid hormone receptor superfamily. He actually compared the DNA binding domains of various steroid hormone receptors like GR, MR, PR, ER, as well as the thyroid hormone receptors VRBA as well as the vitamin D receptors, and then demonstrated that extensive homology exists in the DNA binding domains of all these receptors, suggesting that all these probably belong to a superfamily of transcription factors known as the steroid thyroid hormone receptor superfamily. And they also shared the same zinc finger DNA binding domain. They are all capable of, at least hypothetically, they are all capable of coordinating the zinc finger, and therefore, these amino acids in the DNA binding domain can actually form a 2 zinc finger domains. But the question is, how do they bind to what kind of DNA sequences they recognize, and how do they bind to DNA, and do they bind to same sequences or do they bind to different sequences? What we now discuss is that despite having homology in the DNA binding domains, despite they all containing zinc finger domains, the sequences which they recognize are quite different. That is what will be the crux of today's talk. So, once people realized that before the advent of molecular biology, researchers were purifying receptors like glucocorticoid receptor, progesterone receptors, and by using techniques like electrophoretic mobility shift assay, DNA zone footprinting, identification of DNA zone hypersensitive sites, etc. Using these techniques, they have been able to demonstrate that genes which are responsive to these hormones contain binding sites for these receptors. For example, genes like promoters like that of mouse mammary tumor virus promoter, human growth hormone, metallothionein, the tyrosine hydroxylase, tyrosine aminotransferase, all these genes are responsive to glucocorticoid hormones, and when they looked at the promoter regions of all these genes, they found that the purified glucocorticoid receptor can actually bind to the promoter regions, and in fact, based on these sequences to which these glucocorticoid receptors bind, a consensus sequence was derived, like for example, GGTACA, TGTTC or AGAACA, TGTTCT, which is an inverted repeat. So, by comparing the sequences to which this glucocorticoid receptors binds to various promoter elements, a consensus sequence was derived for the binding of glucocorticoid receptor. Similarly, in the case of estrogen responsive genes, by analyzing the promoter regions of the estrogen responsive genes and studying by using techniques like EMSOL, DNA zone footprinting, etc., by identifying exact binding sites of the either the purified receptor, or once you have a cloned receptor, you can overexpress the receptor in the E. coli cells, or you can use receptors which are translated in vitro using rabbit reticulocyte lysates, and using these receptor preparations, people have been able to demonstrate they identify exact binding sites for estrogen receptor as well, which is GGTCA 3 nucleotide spacer TGACC. Now, but once you demonstrate that these receptors actually can bind to specific response elements, then the question comes, when they bind to the response elements, do they really activate transcription? For this, a very well designed assay called as cis-trans co-transfection assay was developed. So, what you do in this assay is that you design 2 different kinds of plasmids. One of the plasmids you suspect, suppose you suspect that the promoter region between -338 and -315 in the vitellogenin gene contains a estrogen response element, and you want to really demonstrate that this region of the promoter, when estrogen receptor binds, it activates transcription. Now, how do you demonstrate that? What you do is that you take this response element and clone it in front of a reporter gene, which can be either a chloramphenicol acetyltransferase, which converts chloramphenicol to acetyl chloramphenicol, or it can be luciferase, which will convert luciferin which can oxidize luciferin resulting in the emission of light, or it can even be reporter genes like beta galactosidase which when the enzyme is made will give a color when you use a chromogenic substrate. So, by linking these response elements with a minimal promoter which only consists of binding sites for RNA polymerase II and general transcription factors, and putting them in front of reporter genes and then when you now express another expression plasmid which I call plasmid A or plasmid B, in this case the nuclear receptor cDNA is cloned in front of an ubiquitous promoter. It can be a promoter from a cytomegalovirus promoter or a Rous sarcoma virus promoter. All these promoters work very well in any eukaryotic cell line, many of them mammalian cell lines. So, when you introduce this cDNA, what we call as a mammalian expression vector, now these cells will start expressing the nuclear receptor, and now the nuclear receptor will go and bind to the response element, and now when you add the hormone, you will now start seeing the chloramphenicol acetate transferase activity. So, I have just shown here. If you want to demonstrate that a thyroid hormone response element actually is present in the promoter region of a certain gene, you take this, you design an oligonucleotide, or you take out this promoter region, put it in front of a reporter gene, and now when you now express the thyroid hormone receptor in the same cell which have been transfected this plasmid, you transfect both the cis plasmid and the trans plasmid. Cis plasmid is the one in which the response element is linked to a reporter gene. Trans plasmid is the one which contains the receptor of your interest. Now the receptor now expressed from this receptor from this plasmid goes and binds to the response elements and expresses the reporter gene, and now you can do what is called as a reporter gene assay. If you use cat as a reporter gene, you can actually measure the cat activity in the presence or absence of a hormone, and this is how a typical result looks like. So, this is chloramphenicol, this is acetylated form of chloramphenicol, and you can see when you make these cell extracts in the absence of hormone, chloramphenicol is not converted to the acetylated form, but when you add hormone, now chloramphenicol is getting converted into acetylated hormone. What it means is that the hormone, the thyroid hormone receptor which has been synthesized from this trans plasmid, bound to the thyroid hormone, gone and then bound to the response element and activate the expression of the cat gene, and this cat is now able to convert chloramphenicol into acetyl chloramphenicol. So, by using this kind of cis trans co-transfection assays, people have been able to demonstrate that binding of the receptor, whether it is a glucocorticoid receptor, thyroid hormone receptor or estrogen receptor, can, when they bind, it can actually activate transcription of a downstream reporter gene when the cognate ligand is present. So, this cis trans co-transfection became a very useful assay for demonstrating that a binding site or a response element, the binding site for a nuclear receptor can actually function as a hormone response element in vivo. So, using these kinds of assay, people have identified the binding sites for thyroid hormone receptors, because the cDNA for thyroid hormone receptor was present. So, you can now take the cDNA for thyroid hormone receptor, put it in a trans plasmid and suspect that we know many genes which are responsible for thyroid hormones, and take those promoter regions, put it in front of this reporter genes and do the cis trans co-transfection assay and see what are the exact binding sites to which the thyroid hormone receptor binds and activates transcription. So, with such studies, people have demonstrated that the thyroid hormone receptor actually bind to short repeated sequences of a DNA called thyroid or treated response elements or TREs, and these thyroid hormone response elements can actually consist of AGG-TCA separated by 4 nucleotides. So, the response element for thyroid hormone or the TRE is nothing but AGG-TCA 4 nucleotide spacer AGG-TCA, and this is called as DR4 because the 2 direct repeats are separated by a 4 base pair spacer. So, a DR4 element is a thyroid hormone response element or a TRE, and you can see very clearly this response element is very different from the steroid hormone response element. For example, if you take estrogen receptors, it is AGG-TCA TGA-CCT. So, it is a palindrome. So, AGG-TCA is actually present on the other strand. So, AGG-TCA, the palindrome sequence, and estrogen receptor binds on the 2 sides of the double strand DNA helix, whereas in the case of the thyroid hormone response element, the half site sequence is same as that of estrogen receptor, but in this case the half site is present as a direct repeat separated by 4 base pair spaces. So, the thyroid hormone receptor, which is a type 2 receptor, binds a direct repeat motif, whereas the type 1 receptor like estrogen receptor and glucocorticoid receptor bind to a inverted repeat or a palindromic sequence, where the 2 half sites are present on as palindromic sequences or inverted repeats. So, an important observation is that the thyroid hormone response element or TRE consists of 2 half sites containing the sequence AGGTCA separated by a 4 base pair spacer, and this is known as direct repeat 4 or DR4 element. So, if a promoter has a DR4 sequence, it is likely to be responsive to thyroid hormone. Thyroid hormone receptor can bind to such sequence and activate transcription. By analyzing a number of other thyroid hormone responsive promoters, people came to realize that not only the direct repeat can present as a direct repeat separated by 4 base pair spacer or DR4, in some of the promoters the same direct repeat can also be provided as inverted palindromes or palindromic sequences, and such variants of these direct repeat elements can also function as a thyroid hormone response elements. So, analysis of other thyroid hormone responsive promoters indicated that the half sites of the TRE can be present either as direct repeats, palindromes or inverted repeats. But to avoid complications, just remember the most important observation that came is that the thyroid hormone receptor actually binds to promoters in which the direct repeats, the AGGTCA motif is present and it is separated by a 4 base pair sequence. A very important discovery in the area of type 2 receptors or the nuclear receptors is the proposal of a rule known as 3-4-5 rule. This is a landmark paper which was actually published by Kazuhiko Umesono, a very brilliant postdoc in the laboratory of Ronald M. Evans at Salk Institute, who unfortunately passed away at a very early age. And he came up and published this paper, which is titled Direct Repeats as Selective Response Elements for the Thyroid Hormone, Retinoic Acid and Vitamin D3 Receptors. These people in the area in this field often consider this a very important paper or landmark discovery, wherein he actually proposed that a very simple rule exists at the molecular level for the binding of vitamin D receptor, thyroid hormone receptor and retinoic acid receptor. Now, what is this rule? What he actually proposed is that he started looking at the binding of thyroid hormone receptor to thyroid hormone response elements, and he found that thyroid hormone response elements consists of direct repeat half sites separated by a 4 base pair spacer. This is what we discussed now. So, AGGTCA, when you separate by 4 base pair sequence, it becomes a thyroid hormone response element. Now, what he now did is that if you now increase the spacing by one more nucleotide, that means instead of 4 base pair spacer, we make it a 5 base pair spacer, thyroid hormone receptor does not bind it any longer. So, the TRE, that is a DR4 element, can be converted into a retinoic acid response element by increasing the spacing between half sites by one nucleotide sequencer. So, the half site sequence is the same, but instead of 4 nucleotide spacer, if you now increase the spacing to 5 nucleotide spacer, a TRE now becomes an RARE or a retinoic acid response element. So, a when a DR4, when it becomes DR5, a thyroid hormone response element now becomes a retinoic acid response element, and by just increasing one nucleotide spacing, you can convert a thyroid hormone response element into a retinoic acid response element. So, by increasing the spacing by one nucleotide, the resulting retinoic acid response element is no longer functions as a thyroid hormone response element. And more interestingly, instead of adding one nucleotide, now if you remove one nucleotide, that means if a 4 base pair spacer now becomes 3 base pair spacer, now it becomes a vitamin D3 response element, or vitamin D receptor can now go and bind to this. Decreasing the half site spacing by one nucleotide converts a thyroid hormone response element to vitamin D3 response element. That means here AGGTCA is separated by 3 base pairs and this now no longer binds to thyroid hormone receptor. So, this study suggested that a simple physiologic code exists in which half site spacing plays a critical role in achieving selective hormone response elements. You can see now at the molecular level how a simple code can lead to different physiological responses. Now, I have vitamin D, which has totally different physiological effects, thyroid hormone, entirely different physiological effects, retinoic acid, which is a morphogen, again the physiological effects are totally different. But at the molecular level, the DNA sequences which these receptors are recognizing and therefore activating specific target genes is just different by one base. So, if we have 2 half site sequence separated by 4 base pair nucleotide, it becomes a thyroid hormone response element. If you have 2 half sites separated by 3 base pair sequence, 3 base pair spacer, it becomes a vitamin D response element, and if these 2 half sites are separated by 5 base pair spacer, it becomes a retinoic acid response element. So, that is why this came to be known as 3-4-5 rule. That means DR3 is a vitamin D response element, DR4 is a thyroid hormone response element, and DR5 is a retinoic acid response element. So, this following the publishing of this paper this entire observation became known as the 3-4-5 rule, where a DR3 is a vitamin D receptor binding site, DR4 is a thyroid hormone receptor binding site, and DR5 is a retinoic acid receptor binding site. So, while the DNA binding specificity of a number of nuclear receptors whose ligands are known, such as glucocorticoid receptor, estrogen receptor, thyroid hormone receptor, vitamin D receptor was being deciphered, efforts were also being made to clone and characterize other nucleotides, other nuclear receptors such as retinoic acid receptors. As I told you, Ron Evans proposed the existence of a steroid thyroid hormone receptor superfamily, and he proposed that in addition to the receptors which bind to known ligands, there may be a number of other receptors for which the ligands is not known. In fact, using the DNA binding domains of the glucocorticoid receptor or estrogen receptor or thyroid hormone receptor as probes, people start screening cDNA libraries under low stringency conditions to see are there other receptor like molecules with the same kind of a zinc finger domain as that of glucocorticoid receptor or thyroid hormone receptors. So, when you do these hybridizations under low stringency, in fact, people pulled out a number of other cDNA clones which also had the same kind of a domain structure as either glucocorticoid receptor or thyroid hormone receptor. But they did not bind to glucocorticoid response element, they did not bind to a thyroid hormone response element, nor they bound to those ligands, indicating that the exact physiological function of these receptors is not clear, because we do not know what kind of sequences the receptor binds and what are the ligands for the receptors. And such receptors are became to be known as orphan receptors. So, orphan receptors are nothing but they are also these receptors share the same structural features as that of steroid or nuclear receptors, but the sequences to which they bind or the ligands with which they interact is not known. So, the exact physiological function of these orphan receptors were not clear. So, a major area of research during the late 80s and early 1990s is to understand what is the function of these orphan receptors, what kind of response elements they bind, what are the ligands for these orphan receptors. So, let us see how these discoveries were made. For example, a cDNA which has very high homology to that of glucocorticoid receptor or thyroid hormone receptor was identified. It had the same kind of a domain structure, and in fact, this receptor was found to be was found to bind to retinoic acid, labeled retinoic acid, suggesting that this can actually be a retinoic acid receptor. But the response element to which this retinoic acid receptor binds was not very clear. So, this is where a very novel strategy was used to identify response elements or the recognition sequences of these orphan receptors. So, what you do is you take advantage of the fact that these orphan receptors have the same kind of a domain structure as the known receptors which have known ligands. For example, if we take glucocorticoid receptor, it has an amino terminal domain, a DNA binding domain, and a ligand binding domain which binds to glucocorticoid hormones like cortisol. And we know the DNA binding domain of glucocorticoid receptor binds to glucocorticoid response elements, right. Now, we have, for example, retinoic acid receptor, and we know the ligand binding domain of retinoic acid receptor binds to retinoic acid, but we do not know what kind of sequences to which the retinoic acid binds. But to actually demonstrate that this indeed is a retinoic acid receptor and it can actually activate transcription when retinoic acid bind, a very clever strategy was used, wherein you introduce restriction sites in the DNA binding domains, like a not one site here, XH1 site here, not one site here, XH1 site here, and now you remove the DNA binding domain of glucocorticoid receptor as a not one XH1 fragment and replace it with the not one XH1 fragment of retinoic acid receptor. And you can actually see this is what is a chimeric receptor is made. So, you replace the DNA binding domain of retinoic acid receptor as a not one XH1 fragment, pull it out, and then put the not one XH1 fragment of glucocorticoid receptor into the retinoic acid receptor. So, this is now called as RGR. Now, this is GGG, this is RRR, and this is RGR. That means it has the amino terminal domain of retinoic acid receptor, ligand binding domain of retinoic acid receptor, but DNA binding domain of glucocorticoid receptor. Now, if you transfect this chimeric receptor into cells, and if you now put a reporter plasmid and glucocorticoid response element, this RGR was now able to activate transcription in response to retinoic acid. So, by using this kind of a domain swapping experiments, people have started demonstrating or screening ligands for these various orphan receptors. So, in this landmark paper, where identification of receptor morphogen retinoic acid, they conclusively demonstrate that when retinoic acid binds to this chimeric receptor, it activates transcription, clearly indicating that the cDNA that they have isolated is actually retinoic acid receptor, although the sequences to which the receptor binds was not clear. at that time. So, using these kinds of strategies, people have started screening a number of orphan receptors to see what kind of ligands, when the receptors bind, can activate transcription. So, basically what you do is you go ahead and make extracts of various liver tissue or brain tissue or during different stages of development, and then fractionate these extracts and then separate these various fractions through HPLCs and take these various fractions and see which of the fractions can actually activate an orphan receptor. So, the strategy, since you do not know what kind of sequences these orphan receptors bind, you use this chimeric approach. So, you take the DNA binding domain of a well characterized receptor like glucocorticoid receptor and put it into the and replace it with the DNA binding domain of the orphan receptor, and this chimeric receptor now introduced to cell lines, and now you add various fractions of tissue extracts and see which fraction will now serve as a contains a ligand for these orphan receptors. So, it became possible to screen or demonstrate or look for biological activity or a receptor activation ligands in various cell extracts, although we have no knowledge of the sequences to which these orphan receptors bind. By using these kinds of studies, people started identifying ligands for various orphan receptors. So, on one hand, people started identifying what kind of response elements to which this binds. On the other hand, since a large number of orphan receptors have been isolated using these low stringency hybridization techniques, people, even without the knowledge of what kind of sequences these receptors bind, by making this kind of chimeric receptors, people started screening for ligands from various cell extracts or synthetic libraries, what kind of molecules can actually bind to the ligand binding domains and activate transcription. So, this became the common strategy for the identification of ligands for a number of orphan receptors, for which we do not know what is the ligand, nor we know what kind of sequences these receptors bind. So, while the DNA binding specificity of a number of nuclear receptors whose ligands are known, such as T or VDR, was deciphered, efforts were also being made to identify ligands of orphan receptors. One such orphan receptor, which later was identified as Retinoid X receptor, is a very important discovery. So, of the number of orphan receptors that were available, one of the receptors was soon identified as Retinoid X receptor. Now, what is this X? Because in the previous couple of slides I actually showed the identification of retinoic acid receptor. The retinoic acid receptor activates transcription in response to all trans retinoic acid, that is the ligand for retinoic acid receptor. But there is another receptor which was called as Retinoid X receptor or RXR. Now, what is the difference between RAR and RXR? The only difference is that when you take the cDNA coding for RXR or cDNA coding for RAR and replace the DNA binding domains with that of glucocorticoid receptor, and now if you add all trans retinoic acid, even at very low concentrations, even at nanomolar concentration, the RAR, the ligand binding domain of retinoic acid receptor, is able to activate transcription. Whereas in the case of retinoid X receptor, it required very high concentrations of all trans retinoic acid, in fact maybe even micromolar concentrations. So, RXR specifically responds to retinoids because high concentrations of all trans retinoic acid could activate RXR alpha, whereas the RAR alpha could be activated by even very low concentration of all trans retinoic acid. So, people suspected that it is not the all trans retinoic acid, but when you add all trans retinoic acid, it is now getting converted to some other isomer, and it is an isomer of all trans retinoic acid which is acting as a ligand for this other variant of RAR, and so we do not know what that isomer of retinoid, it was called as X. Since this isomer is different from the all trans retinoic acid, it is called as RXR or some derivative of retinoid, which is the ligand for this receptor. And soon this retinoid X was found to be an isomer of retinoic acid called 9-cis retinoic acid. An isomer of all trans retinoic acid called as 9-cis retinoic acid was found to be the ligand for RXR alpha, as well as two other related subtype calls RXR beta and RXR gamma. So, you can see how using this kind of chimeric approaches, people started identifying ligands for orphan receptors. So, you do not know what kind of response element this RXR binds, but by replacing the DNA binding domains of RXR with that of another one, and by screening various synthetic derivatives of all trans retinoic acid, people came to know that a 9-cis retinoic acid is actually the ligand for this particular receptor, and therefore, the retinoid X receptor is actually the receptor for 9-cis retinoic acid. So, the retinoic acid receptor is the activates transcription response to all trans retinoic acid, retinoid X receptor activates transcription response to 9-cis retinoic acid. So, this is just an example to show how ligands for orphan receptors were identified, although even without the knowledge of the exact sequences to which these receptors actually bind. So, while in the 1987 two groups, one from Branavan's loop and one from Schambon's group actually demonstrated the cloning of the retinoic acid receptor, and it took about 3 years to actually clone the retinoid X receptor and actually demonstrate the retinoid X receptor is the receptor for 9-cis retinoic acid, whereas the retinoic acid receptor which was cloned in 1987 is the receptor for all trans retinoic acid. So, two major isomers of retinoic acid, you have all trans retinoic acid which binds to RAR and activates transcription, another isomer of retinoic acid 9-cis retinoic acid binds to RXR and activates transcription. So, these are again two landmark papers. Now, you also brought the retinoic acid into the realm of nuclear receptor superfamily, and then say that in fact there are 3 isoforms of RAR called RAR alpha, beta and gamma. They are expressed in different tissues, meaning that there could be variations in these receptor cell types. Similarly, the RXR also, they found out there are 3 different isoforms of RXR, alpha, beta and gamma, and so on and so forth. So, using these kinds of a chimeric receptor strategy, people started screening libraries of compounds and see whether these compounds are made from various tissue extracts or cell extracts, or it could be synthetic libraries made by organic chemists. And by using this kind of chimeric approach, people started screening libraries of compounds to identify what is the receptor for all these orphan receptors. And another important landmark in the area of type 2 receptor research is that the RXR not only was the receptor for 9-cis retinoic acid and activate transcription response 9-cis retinoic acid, it also became clear that this RXR also is very important for the binding of other nuclear receptors like thyroid hormone receptor, vitamin D receptor and retinoic acid receptor. Now, how was this discovered? Now, once the cDNA for thyroid hormone receptor, vitamin D receptor and retinoic acid receptor were cloned, when they take the cDNAs and do what is called as an in vitro transcription and translation. Now, you can now take the cDNA, put it into a expression vector, put it into a vector which is now controlled by T7 RNA polymerase. Now, if you add T7 RNA polymerase, you can make large amounts of this RNA. So, you have RNA coding for either vitamin D receptor, thyroid hormone receptor or retinoic acid receptor. Now, if you now take this RNA and put it in rabbit reticulocyte lysates, you can make a protein. Now, if you take, these are called as in vitro translated proteins. Now, if you take such in vitro translated proteins and then do what is called as an electrophoretic mobility shift assay with a suspected thyroid hormone response element or vitamin D response element or retinoic acid response element, these in vitro translated proteins could not bind. That means, either an in vitro translated thyroid hormone receptor or a receptor that was made in E. coli cells, okay, they did not actually bind to thyroid hormone response elements. But when you take a nuclear extract from, so for example, CV1 cell, any eukaryotic cell lines, and add little bit of this extract to this in vitro translated receptor or bacterial express receptor, it bound very well. So, these kind of studies very clearly mentioned, suggested that for the vitamin D receptor or thyroid hormone receptor or retinoic acid receptor to bind to the response element, there is some other factor that is required. So, some factor which is present in these cell extracts is actually essential for the efficient binding of thyroid hormone receptor or vitamin D receptor or retinoic acid receptor to the response elements. So, people started hunting what is this factor that is present in cell extracts that makes these receptors bind very efficiently to their response elements, and soon this factor turned out to be retinoid X receptor. So, you can see the retinoid X receptor not only activates transcription in response to 9-cis retinoic acid, it also is very essential for the binding of thyroid hormone receptor, vitamin D receptor and retinoic acid receptor. So, it became very clear, in order for the vitamin D receptor to bind to a DR3 element or a vitamin D response element, you require RXR. And by using a number of studies from a number of laboratories, it became very clear of the 2 half sites which is present, the vitamin D response element separated by 3 base pair spacer, the RXR bind to the 5' half site and the vitamin D receptor binds to the 3' half site. So, the RXR thyroid hormone, the RXR VDR heterodimer is the one that can bind to this DR3 element, whereas vitamin D receptor alone cannot bind. Similarly, the RXR in combination with thyroid hormone receptor heterodimer can now bind to a DR4 element or a thyroid hormone response element, whereas RXR and RAR heterodimer can bind to a RARA DR5 element. So, you can see the RXR became the common heterodimeric partner for vitamin D receptor, thyroid hormone receptor and retinoic acid receptor. So, again it is a very important observation in the area of nuclear hormone receptor research. Soon people started looking at, as I said, number of orphan receptors was clean. So, like we began this discussion with steroid hormone receptors, then we have now discussed about retinoic acid receptor, thyroid hormone receptor, vitamin D receptor. At the same time, people also started looking at steroid hormones like ecdysone in the case of insect cells, and they found that ecdysone receptor is also has the same structure as that of steroid hormone receptors, and people started identifying ecdysone response elements in the insect promoters, and the ecdysone receptor also became a member of the nuclear receptor superfamily. And many other insect receptors were discovered later, like ultraspiracle and so on and so forth. And soon a number of other, today actually we know that there are a number of other orphan receptors like LXR, which is liver X receptor, farnesyl X receptor, pregnein X receptor and so on and so forth. All these which were actually are known as orphan receptors in the late 1980s and early 1990s. Now people have identified what are the ligands for all these receptors. So, orphan receptors which were classified as orphan receptors in the late 1980s and 90s became full-fledged members of nuclear receptors once the ligands for these receptors were discovered and also the response elements to which these receptors were identified. So, they all have very important physiological functions, and today one of the major areas of research is to see what are the physiological functions of these receptors and what are the, can we develop agonists or antagonists against these nuclear receptors, and we will discuss some of these things and see how understanding the structure and function of nuclear receptor can now lead to development of number of drug molecules which can either be agonists or antagonists of receptor of these nuclear receptors, and they have very important biomedical applications. So, I have listed here a number of receptors were actually discovered: HNF4, COOP-TF, chicken warm upstream promoter, transcription factor, nerve growth factor, induced build protein, NGF-IB and so on and so forth. But what I am trying to tell you is that the story which started with the cloning of steroid hormone receptors led to the discovery of number of other receptors which had same structural features, and a major activity was initiated in the area of nuclear receptors to see what kind of response elements to which these receptors bind and what are the ligands for these various receptors. And soon it became clear the 3-4-5 rule actually became 1-2-5 rule or 1-2-3-4-5 rule, because RXR was found to be a heterodimeric partner not only for binding to receptors for thyroid hormone receptor and the TR, VDR and RAR, it also was required for other receptors. Now, what are these? The direct repeats of AGG TCA with variable spacing was now found to serve as binding sites for different nuclear receptors which have RXR as a common heterodimeric partner. And so far I told you that for DR3, DR4 and DR5, vitamin D receptor, thyroid hormone receptor, RAR to bind to these elements you require RXR. But soon it became clear that the RAR RXR heterodimer can not only bind to a DR5 element, but it can also bind to a DR2 element, that is AGG TCA separated by 2 base pair sequence. And soon another receptor called peroxisome proliferated activator receptor, PPAR, was discovered, and again the PPAR again was found to bind to a DR1 kind of a sequence in heterodimer along with the heterodimer of RXR. And in fact, people have actually shown RXR itself, in order to activate transcription in response to 9-cis retinoic acid, has to bind to promoters containing a DR1 kind of a motif. So, you can see RXR not only serves as a heterodimeric partner for a number of other receptors, it can also bind to its own response elements as homodimers containing a DR1 kind of a sequence and activate transcription in response to 9-cis retinoic acid. So, RXR became a very unique receptor among all the receptors discovered so far, in the sense that it has the quality of both steroid hormone receptors and type 2 receptors. I told you steroid hormone receptors primarily bind as homodimers, type 2 receptors primarily bind as heterodimers. But RXR is one unique example where it can not only bind as heterodimers with other nuclear receptors, it can also form homodimers and activate transcription with specific response element. So, RXR is a very important discovery in the area of nuclear hormone receptors. And in all these heterodimeric binding, the RXR binds to the first half site or the 5' half site, and the partner, whether it is thyroid hormone receptor, vitamin D receptor, RAR, binds to the 3' half site, and that is how they bind and then activate transcription. So, this is what is known as the 3-4-5 rule, which was discovered by Kaz Umisono, soon became a 1-2-5 rule because 2 more response elements DR1, DR2 were discovered, which also requires RXR as a binding partner. So, this is the summary of all the research that went on into this to actually show RXR RXR homodimers can bind to promoters containing a DR1 kind of a motif, where AGGT CAGGT is separated by one base pair rule, one base pair spacer. In addition to RAR RXR, RXR can also form a heterodimer with RAR, another receptor called peroxisome proliferator activated receptor, hepatocyte nuclear factor 4, chicken ovalbumin upstream promoter transcription factor. So all these receptors along with RXR as a heterodimeric partner bind to promoters containing a DR1 kind of a response element. Whereas RXR PPR heterodimer as well as RXR RAR heterodimers can also bind to promoters containing a DR2 kind of a sequence, where the two half sets separated by two base pair spacing. Whereas the DR3 response element is a unique response element for RXR VDR vitamin D receptor heterodimer, whereas the DR4 type of response element is the response element for RXR thyroid hormone receptor, and later the same DR4 sequence was also found to be recognized by RXR LXR heterodimer, and also another novel receptor called as CAR. And the DR5 response element, where the two half sets are separated by 5 base pair spacer, become the response element for RXR RAR as well as another transcription factor called NGF5B. So you can see how RXR became a common heterodimeric partner for a number of other receptors, and by binding to different response elements separated, where the two half sets separated by different lengths of spacer, different physiological responses can be brought about. So the summary of what we have discussed so far: the steroid hormone receptor binds to inverted repeat sequences separated by 3 base pair sequence, and we had a number of other receptors that we have discussed here, where the two direct repeats are separated by either 1, 2, 3, 4 or 5 base pair spacers, and if it is separated by 1 base pair, these are the receptors which go and bind. If it is separated by 2 base pair spacer, these are the receptors which will bind in combination with RXR. Similarly, the receptors indicated in red here, along with the common heterodimeric partner, can bind to different direct repeat sequences. So the RXR, in addition to binding to 9,6 retinoic acid and regulating the expression of genes through DR1 element, is also the key heterodimeric partner for a number of other nuclear receptors. This is the paradigm that emerged out of all these studies. So RXR became a very important molecule along with the various nuclear receptors. So today we have a number of nuclear receptors, and this just shows you some of them which have been very well characterized. The panel here actually shows the steroid hormone receptors. You have MR, mineralocorticoid receptor, progesterone receptor, androgen receptor, glucocorticoid receptor, estrogen receptor, and so on and so forth. Again, you can see all these receptors have the same structural features. You have an amino terminal domain, you have a DNA binding domain, and you have a transcription activation domain. Amino terminal domain, DNA binding domain, and transcription activation domain. The amino terminal domain is supposed to encode what is called as a ligand independent transcription activation function, therefore it is called as TF1, whereas the ligand binding domain encodes what is called as a ligand dependent transcription activation function. In the next class we will discuss exactly how the ligand actually activates transcription, how the ligand binding to the ligand binding domain results in the activation of transcription. For example, FXR or the foreign SLX receptor, the ligand actually was found to be bile acid metabolites. The RXR, as I said, 9,6 retinoic acid is the ligand for RXR. Many oxysterols were found to be ligands for liver X receptors. Peroxisome proliferator activated receptor, a number of fatty acids and eicosanoids were found to be ligands for the PPR group of molecules, and all these are very important targets for a number of drugs now, for a number of diseases like arthritis, diabetes, and so on and so forth, obesity. All these receptors seem to play a very important role in activating the expression of genes involved, and if you have improper activation, it results in diseases like obesity, diabetes, and so on and so forth. So there are very important molecules as far as drug targets and drug discovery is concerned. Vitamin D receptor and thyroid hormone we already. So the story started from glucocorticoid steroid hormone receptors, led to the discovery of retinoic acid receptor, thyroid hormone receptor, vitamin receptor. Now we have peroxisome proliferator receptor and many other receptors which are binding to a number of important molecules like bile acid metabolites, oxysterols, fatty acids, eicosanoids. So all these molecules seem to be activating expression of target genes by binding to specific intracellular receptors, and they all together constitute the nuclear receptor superfamily. So this is just a summary of what we discussed so far. I will not discuss again, just to tell you that the steroid hormone receptor type 1 receptors bind as homodimers to inverted repeat sequences like GREs and EREs, and the only 2 base pairs distinguish between a GRE and a ERE. And the type 2 receptors what we discussed today consists of nuclear receptors like RXR, peroxisome proliferate activated receptor, vitamin D receptor, thyroid hormone receptor, retinoic acid receptor. All these things, the RXR serves as a common heterodimeric partner. This carton is a little bit wrong. Usually the RXR binds to the 5' half site and the nuclear receptor binds to the 3' half site. So these are pretty different from the type 1 receptors which primarily bind as homodimers, and while these receptor binds to inverted repeats or palindromic sequences, these type 2 receptors bind to direct repeat sequences separated by spacers which can range from 1 to 5. I have again summarized. The steroid receptors are primarily cytoplasmic in the absence of ligands, and the DNA binding is ligand dependent, whereas the nuclear receptors are already nuclear. They do not associate with heat shock proteins. They can bind to DNA either in the liganded or unliganded form. Steroid hormone receptors primarily bind to DNA as homodimers. Nuclear receptors primarily bind as heterodimers with the exception of RXR, which can bind both as homodimer as well as heterodimer. While steroid hormone receptors primarily bind to inverted repeat sequences or palindromic sequences, the other nuclear receptors we discussed today primarily bind to direct repeat sequences. This is again a summary of what we discussed, clearly showing that a number of other receptors, in addition to what we discussed today, a number of other receptors have been discussed and their response elements to which they bind also have been characterized, and all these receptors, the common heterodimeric partner is RXR. So this is the summary of nuclear receptor subfamily. Now a new nomenclature was evolved to make sure that there is no confusion. For example, estrogen receptor alpha is now designated as nuclear receptor 3A1. Glucocorticoid receptor has been designated as nuclear receptor 3C1 and so on and so forth. A new nomenclature was developed to make sure that everybody follows the same nomenclature, and what we discussed so far in the last two classes is the thyroid hormone receptors, retinoic acid receptors, estrogen receptors, the steroid hormone receptors and RXRs. What I have shown in the red, little bit of PPR I just mentioned, but there are a number of other receptors and lot of research is going on to see what kind of ligands they bind, what kind of response elements they bind, and what is the physiological response of these genes. And what I have indicated here as arrows, all these receptors which are indicated by arrows, they are all very important drug targets. So I think I will stop here. This just to tell you that the research which started with purification of vitamins, thyroid hormones ultimately led to cloning of nuclear receptors, and how it became a very important field. And what we will discuss in the next class is, what we discussed so far is how this receptor binds to DNA, and what we will discuss in the next class is that once they bind to DNA, how do they activate transcription, how do they interact with various transcription factors and activate transcription. And these are some of the references, key references I have indicated here. You can go through, and then there is also a very important website where you can go, and there are nice videos which are actually played to give you updates of what is going on in this area. It is called as Nuclear Receptor Signaling Atlas. I think I will stop here.